Welcome to the EvoEd COVID-19 video series. In this video, we'll take a look at the genome of SARS-CoV-2. Then, we'll discuss how the virus can mutate and how natural selection may lead to the emergence of new variants. By the end of this video, you should be able to describe the major features of the SARS-CoV-2 genome and spike gene, and explain which features make it likely for new variants of the virus to emerge. Viruses vary in their morphology and size, depending on their mode of transmission and the organisms they infect. However, viruses are classified by the structure of their genetic material. Generally, this classification is based on whether their genome is stored as DNA or RNA, and whether it is double or single-stranded. Retroviruses, which have their own classification, work a bit differently by integrating their genome directly into the genome of their host. Regardless of the type of genomic storage, all viruses work by eventually transcribing their genome into mRNA that can then be translated by the host cell into viral proteins. SARS-CoV-2 and other coronaviruses are classified as positive-sense single-stranded RNA viruses. This type of RNA must be transcribed into negative-sense RNA, which can then be used as a template to transcribe mRNA. Here is the entire genome of the first SARS-CoV-2 strain that was initially identified in Wuhan, China. It contains about 30,000 nucleotide base pairs across 11 genes that code for 12 potential protein products. Within the genome, we're specifically going to focus on the spike gene and the protein it codes for. The spike protein is comprised of 1,273 amino acids and is coded for by a sequence of 3,821 nucleotides. It's comprised of two major structural subunits and has a binding domain that is specialized for the ACE2 receptor. The reason we'll focus on the spike protein is because its function plays a really big role in the virus's ability to infect cells and to replicate. Firstly, the spike protein influences the ability of the virus to bind to the receptors of cells and to enter them. Secondly, the spike protein is one of the major molecules that our immune system targets when trying to eliminate the virus from our bodies. So, any mutations that alter the spike protein's ability to bind and enter the cell or to evade the immune system will have a positive impact on that virus's ability to replicate. Just like in living things, mutations arise randomly in viruses during the process of DNA or RNA replication. However, the rate of mutation varies across organisms. Researchers have identified a very interesting negative association between genome size and mutation rate across organisms. This means viruses like SARS-CoV-2, which tend to have really tiny genomes, also tend to have high mutation rates. The fact that coronaviruses have such a high mutation rate and are able to replicate very quickly is the perfect recipe for new variants to emerge. To understand why this is, let's review the three conditions required for evolution by natural selection. First, individuals in the population must vary in their traits. Second, at least some of this variability is heritable, or in other words, has a genetic basis. And third, some variants must have greater reproductive success compared to others. So in the case of SARS-CoV-2, the high mutation rate and high rate of replication means there will be lots of heritable genetic variability amongst the spreading viruses. Now, although many of these new mutations may reduce the ability of the virus to replicate, viruses harboring such negative mutations will quickly die out. But any of the variants that increase the rate of viral replication due to increased transmissibility or the ability to evade adaptive immunity will rapidly spread throughout the population and become the dominant variant. So, in other words, to walk through this process from start to finish, first, mutations may randomly arise that change the shape of a spike protein in a given virus. Now, some of these changed spike proteins might result in viruses that are either more transmissible or better able to evade the immune system of their host. Now, viruses with these traits will then replicate in greater numbers and spread throughout the population, and over time they may become the dominant variant. Along with other mechanisms, these forces are what drove the evolution of the Delta and Omicron variants, and they'll continue to drive the evolution of new variants as the pandemic moves forward. In the next videos, we'll take a direct dive into the genetics and evolution of the Delta and Omicron variants, and discuss what the future may hold for the evolution of SARS-CoV-2. Well, that's it for this video, and we'll see you next time.